Hi, welcome back to learning about protein synthesis. So, how do we get from the incomprehensible Spanish recipe to some yummy churros? First, the DNA in the cell must be transcribed into pre-mRNA or pre-messenger RNA. This is called transcription, and it occurs in the nucleus. In order to do this, transcription has three stages: initiation, elongation, and termination. In initiation, RNA polymerase, an enzyme that synthesizes RNA, must recognize a section of the DNA called the promoter region to bind to in order to start transcription. In eukaryotes, transcription factors, a group of proteins, first recognize the TATA box, a special string of thymine and adenine. When the transcription factors and the RNA polymerase attach to the TATA box, it is called a transcription initiation complex. Next, in elongation, RNA polymerase adds RNA nucleotides to the 3' end of the DNA, so the RNA is being synthesized from 5' to 3'. Last, in termination, the RNA polymerase transcribes the termination sequence, which is AAU, AAA. Now, the pre-mRNA is complete. The pre-mRNA is like a word-for-word translation of the Spanish recipe into English. Before we use the recipe, it needs to be edited slightly so that we can make the churros easily. Next, the pre-mRNA must be processed to make the actual messenger RNA. To do this, 3 steps occur: a 5' cap, a poly-A tail, and the removal of introns. At the 5' end of the pre-mRNA, a cap of modified guanine nucleotides is added to make sure that it can attach to the ribosome in the next step, but you'll hear more about that later. At the 3' end of the pre-mRNA, a tail of adenine nucleotides is added to protect the mRNA from getting degraded by enzymes. Most importantly, a complex called a spliceosome, as the name suggests, removes unnecessary regions of the pre-mRNA called introns. This spliceosome is made up of SNRPs, small nuclear ribonucleoproteins. Now we have mRNA. Removing these introns allows the exons, the expressed sequences, to leave the nucleus to be translated into protein. Now we have our recipe. Finally, we need to make the churros. The mRNA must be translated into a polypeptide. In this process, there are 3 different types of RNA that are required: messenger RNA, ribosomal RNA, also known as the ribosome, and transcription RNA. Each tRNA has 2 different ends, one with an amino acid and the other with an anticodon that complements the amino acid it carries. First, we start with the ribosome. Each ribosome has a binding site for mRNA and 3 different binding sites for tRNA, called the A, P, and E sites. Translation has 3 stages as well: initiation, elongation, and termination. In initiation, an mRNA strand, starting with the 5' cap, binds with the ribosome. The codon AUG is known as the start codon, and when it is positioned correctly, translation can start. In elongation, different tRNA molecules bind their anticodon end with the triplet codons that match in the mRNA sequence. This occurs in the A site on the ribosome. Then, the tRNA moves over to the P site, where the polypeptide sequence starts to grow as the amino acid forms a peptide bond with the previous amino acid. After this, when another tRNA molecule binds onto the A site of the ribosome and moves over to the P site to add to the polypeptide sequence, the previous tRNA moves to the E site and leaves the ribosome. Just remember, A stands for amino acid, and it's the first step. Then P stands for polypeptide, where it accumulates, and E stands for exit. Lastly, in termination, the ribosome will reach one of the 3 stop codons, UAA, UAG, or UGA. This allows for the polypeptide chain to leave the ribosome and the mRNA to break down. Then, this polypeptide chain is free to form the complex intricate protein shape it is designed to be and carry out major functions in the organism. Now, our churros are complete.
Thanks for watching! If there's a certain part you didn't understand fully, feel free to watch sections of the video again to make sure everything is clear. Hopefully you learned something new today.